right, everybody, buckle up. We are diving deep, deep into the world of genetics today.
That's right, we're talking DNA, the instruction manual for life itself.
I love that, an instruction manual. So if we think about DNA that way,
It's not just any old instruction manual. This thing is packed with information.
more than just how to assemble IKEA furniture, right?
Way more.
Yeah.
We're talking a blueprint for everything that makes you you.
Okay, so before we get too far, before we start building, let's make sure everyone has the basics.
Of course.
What exactly is DNA? If you were trying to explain it to, I don't know, like a 5-year-old?
Well, you could start with the classics. You know, that twisted ladder image,
Yeah.
the double helix.
Yeah, everyone's seen that.
Right? So imagine the sides of that ladder. Those are made up of sugar and phosphate molecules.
Okay, the sides, the backbone.
Exactly. Now, the rungs, that's where it gets even cooler. Those rungs are made up of these things called bases.
So it's not just a random ladder, there's a code built in.
Precisely. 4 bases. Adenine, thymine, cytosine, and guanine.
We can just call them A, T, C, and G, right?
Absolutely. Much easier.
So these 4 letters, that's our genetic alphabet.
Exactly. And just like how we string together letters to form words,
Yeah.
these bases, they form genes.
And those genes are what? Like instructions for our bodies.
Exactly. They determine everything from your hair color to your height to even your risk for certain diseases.
So DNA is the instruction manual, the genes are the sentences, and those 4 bases are the letters. Man, that's a lot of information packed into something we can't even see.
That's the amazing thing. If you stretched out the DNA in just one cell, it would be about 6 feet long.
6 feet. And how many cells are in the human body? Like a gazillion?
Trillions. And each one has a copy of your DNA.
Okay, hold on. My brain is trying to wrap itself around this.
It's mind-boggling, isn't it?
Okay.
And that DNA is constantly replicating itself.
Replicating.
Copying. Every time a cell divides, it needs a full set of DNA, right?
Right, so it can't be missing pages from the instruction manual.
Exactly. So the DNA has to copy itself. And that's where this awesome enzyme, DNA polymerase, comes in.
DNA polymerase.
It's like a tiny copy machine, making sure that new cells get a perfect copy of the DNA.
I'm already picturing this little machine zipping along the DNA strand.
Right. And it's amazingly accurate, but nothing's perfect. And sometimes mistakes happen.
Those are the mutations we hear about.
Exactly. Mutations can happen during this replication process, or they can be caused by things like UV radiation or certain chemicals.
So our DNA is constantly under attack.
You could say that, but our cells are pretty incredible. They have these amazing mechanisms for repairing DNA damage.
DNA repair. So like our cells have their own little mechanics standing by.
Pretty much. For minor damage, like a single base being damaged, there's base excision repair and nucleotide excision repair.
Okay, that sounds complicated.
They are, but think of it like this. Let's say you're proofreading a document. You find a typo. You just replace the wrong letter with the right one, right?
Yeah, easy fix.
That's basically what these repair mechanisms do. They remove the damaged base and insert the correct one.
Our cells are proofreading their own instruction manuals. That's wild. But what about more serious damage?
Like if you spill coffee on that document.
Yeah, more than just a typo, what happens then?
For more severe damage, like a double-strand break, where the DNA molecule actually breaks in two.
Okay, now we're talking major issues.
The cell brings out the big guns. Homologous recombination or non-homologous end joining.
Whoa, 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 slow down there. What are those?
Think of homologous recombination like having a backup copy of your document.
Smart. Always back up your files, folks.
Right. So the cell uses a similar, undamaged DNA molecule as a template to repair the break.
So it's like copying from a friend's notes.
Exactly. Non-homologous end joining, on the other hand, is a bit more of a last resort. It's faster, but not as accurate.
So it's like trying to glue that coffee-soaked document back together.
Kind of. It directly rejoins the broken ends, but it can sometimes lead to small errors.
Ah, so it gets the job done, but maybe not perfectly. Man, it's incredible, though, how our bodies just do this. Like, we don't even have to think about it.
It really is remarkable, and scientists are still working to fully understand these complex processes.
It makes you think. All this from those 4 little letters.
It's like a symphony of molecular processes all happening inside us right now.
It's mind-blowing. The more we learn about DNA and genetics, the more questions we seem to have.
Exactly. And those questions drive new discoveries. It's an exciting time to be studying genetics, that's for sure.
I have a feeling we'll be needing to do a part 2 on this topic.
I think you're right. There's so much more to explore.
But for now, I think our brains need a little break.
A little DNA replication never hurt anyone.
Well played. All right, everyone. Thanks for joining us on this deep dive.